better cells in our body and AI and combining by getting the RBC image analysis. So here is the basic outline for what we will present today. First, I will go through about the introduction, and in the introduction, we will discuss that what was the motivation and what were our goals and challenges we were facing that we decided to set up this AI module system. And then the methodology we would use that, what methodology we would use that, and what the result detection what we got, and then in the end we will shortly give the demonstration about how it works in the real time. So if I talk about what was the goals and the problems in the medical field, or you can say the RBC image analysis in the medical laboratory field, already we know that there are traditional RBC image analysis going on in the hospitals and in the laboratories, but there are some drawbacks. Like it is time taking and it takes a lot of time, and it also needs a specialist to take care of and analyzing the RBC image analysis. But it may also have the drawbacks and disadvantages, like may have a lot of workload of the patients, and there are also chances of the human errors. When we are doing the step-by-step in the laboratories by using the different automated machines, and it takes a lot of heavy equipment and the machinery. So a big factor is also that when we see by manually or by in the hospital, there are a problem about while doing the microscopic image analysis, that is about the focus on the image and recognition of the images and the light contrast problems with the microscope, and this type of problem we face. And also this project, what we got our idea to make this module for set up a system to get the real-time point of care testing and the real-time evaluation. Although there are also automatic machines, but our goal is to focus on the POCT, like point of care testing. That means that we can use in the future at the patient's side and also in the remote areas, and also so it will take less time, and it will give the more accessibility to analyze the RBC image analysis and the patient's condition. So it will give the quick decision-making power for the medical staff to start the treatment for the patients. So it will also save the time, and it's less expensive. So, and then the software we trained for this module is the YOLO V12 we use for, and it is more accurate and it gives the fast and the quick results that also supports our idea of the real-time evaluation, because it gives the more accurate results and the image analysis more quickly and very fastly. So it differentiates between the normal and abnormal RBC, as we know that in our blood, in the patients, there are also the normal and abnormality. So it will give the quick result analysis between the normality and abnormality of the RBC, where the cell is shaped and their image morphology. So it is also the user-friendly, like even the non-technical person can use in case of emergency, or if there is take time to reach the hospital or in the medical laboratory. So before image processing, the method that we use is the median filtering. In this method, the median filter method, this works on replacing the central pixel with the median value of its neighboring pixel. You can see in the both pictures that here first we got this picture and then we use the median filtering so that you can see the background is more clear and it enhances the image quality, so that our AI system can detect it very quickly and give the fast results. And then before image processing, we have the three main key steps. That the first one is the image conversion into the gray background, and then applying the median filtering, and then converting into the finalization. So why we converted the pictures into the gray background? Because in this way the background will be more clear, and while preserving the important details of the red blood cells, image, RBC, and it will preserve the RBC important details of the red blood cells and preserve the cell shapes. Like you can see here, this is the normal RBC. Like you can see it is round shape, and these are abnormal RBC. You can see it is not. It has the spiky edges. So when we apply the median filtering, it enhances the more image quality so that it preserves the details inside the cell and also outside the cell because it cell have the cell membrane. So this process also maintains the more image quality. And then we convert it into the binarization. So binarization convert the image into the just black and white color instead of the colored picture. As we know that we use analysis in the colored picture, but for AI training module, we convert it into the binarization to distinguish between the background and the cell, what actually the cell is. So why we use this software? Because there are a lot of more variants for the YOLO V12, but this system is more faster than other variants because it is more accurate and give the fast and the quick results. So our main focus is to just achieve the real-time evaluation. So it is very easy to use for the non-users, non-technical staff. It don't require any medical staff, hematologist, or any other doctors to specialized staff to use this. So that its interface speed is very fast compared to the other variants. So it supports our idea of setting the goal like POCT, point of care testing, and the real-time evaluation. As I told earlier that this software is more accurate and gives the fastest results just by adding the data and selecting the AI module, and then very fastly give the results in the meantime. Also differentiate the small and the overlapping red blood cells. Here is the main reason that why we use this, why we thought about this project, because in the blood cell diseases, there are a lot of type of red blood cells. In the abnormality, there are many different types and the shapes of red blood cells. So the main focus is to differentiate between the normal and the abnormal in the meantime and the very quickly. So it even give us results small and overlapping red blood cells instead of single cells also. So for the training processes, before training our module, we saw that while giving the training, and then we saw the validation losses box like classification EFL. Here we saw the graph gradually decreases, but it was taking time to give the stable and the quick results. Then in the right side of the picture, you can see that we also saw the evaluation metrics such as precision, recall, and the mean average precision. That you can see it increases gradually and reaches the point 1.0. So in these pictures, you can see that we got our results near 1.0. It means it supports our results RBC image analysis according to our main goal. So we got these results by doing these approximately 300 training cycles. And this is the initial view of our system interface. First we set up the GUI-based design for the initial for the interface analysis. This view allows the selection of the image, and then we can detect the AI module, and we can start and get the results by just few clicks. So our system is real-time evaluation give results, and it is already demo ready. So we can say it also supports our idea for getting the real-time evaluation. Here you can see in the right side the picture. These are the results display, how we got these results. These are the different color boxes. The yellow color boxes show that these are the normal cells, and the cyan and the purple boxes show the abnormal and the overlapping cells. So you can see this is the real-time count of the RBC. So you can see here the total number of the cell count, abnormal cell count, and anomaly ratio is also how much is the normal and how much is the abnormal percentage. You can see here the percentage anomaly ratio and the real-time cell count for this picture. And here's quickly the short demonstration of how our system works.